So let's go through the previous year, last year, that is 2025 CBSE chemistry question paper from the chapter of biomolecules. So this include MCQs as well as descriptive questions, and not in a particular order. Randomly, I have distributed the question paper. So in 2025 CBSE paper, there are actually total 6 sets are there, each set having around 33 questions. So among that, one of the high priority chapter is biomolecules. At least in every test, you will be getting at least, I can say, 8 to 10 marks of question, or at least 9 marks of question or 7 marks of question are coming from this chapter. So if you know the basics of this chapter, it will be very beneficial for you to score good marks. So let's start this particular discussion. So first question is glucose on reaction with bromine water gives. So first of all, only confusion which you will be having in this particular question is either it will be gluconic acid or saccharic acid. So if you know that any of this can be there, you can eliminate the remaining two options. All right, so glucose we can represent like CHO and CHOH 4 times and followed by CH2OH. So when you are reacting this with bromine water is a weak oxidizing agent. So weak oxidizing agent means this will oxidize that particular group which is very easy to oxidize. So here actually we have CHO group as well as primary alcohol. But primary alcohol is comparatively difficult to oxidize when compared to aldehyde. So what will happen is it will only oxidize aldehyde, forming a compound like this. So this compound is called gluconic acid. So why this confusion comes with saccharic acid is because there is another reaction when you are treating this glucose with concentrated HNO3, which is a strong oxidizing agent, which can oxidize both primary alcohol as well as this aldehyde group. So you will be getting COH, CHOH 4 times as well as COH, and this compound is called saccharic acid. So here simple way to remember this one is any question which will come, treatment with bromine water means you need to remember that bromine water actually helps to prove that glucose has aldehydic group. So if the question is about the concentrated HNO3, the answer will be saccharic acid. But since it is about the bromine water, the answer will be gluconic acid. So bromine water is a weak oxidizing agent, so it will only oxidize the aldehyde group. And concentrated HNO3 is a strong oxidizing agent, so it will oxidize both the aldehyde as well as the primary alcohol. So now when it goes to the... So for next question, this is a descriptive question. So in this question, give the point of difference between the following. That means starch as well as cellulose. So if you think about this, starch is something which is found in most of the food items which we are eating, and cellulose is present in plants. Basically, plant cell walls are made up of cellulose. So they both are made up of glucose only. First of all, we can say what you need to write here. I'm only giving the explanation. As per the official answer key, you need to write just one point. I mean, what is the differentiating key point here? So we can write a single thing here. Starch is made up of alpha D glucose units, while cellulose is made up of beta D glucose units. So this is the key point. Though see, this question is basically about the 2 marks. So 2 marks question, you need to be very concise in whatever you are writing. So rather than writing a random words like starch is something found in these food items and cellulose is present in plant walls, that is a very vague way of describing. So when you are writing like this, you should mention what exactly is the key difference between both of them. That means starch is made up of alpha D glucose and cellulose is made up of beta D glucose unit. So be precise when you're writing. So in this B part, we need to write the difference between primary and secondary amino acids. So here also in the official answer key, they have written or any other suitable difference, you can write it. But better to go with what is the most understandable as well as the most clear answer there it is. So the clear answer to this particular question is that primary structure of protein is basically made up of long chain of amino acids. Okay, so primary structure is basically made up of amino acids linked with each other in a specific sequence. And when it comes to secondary structure, so this is a concise as well as what exactly you need to write for the primary structure. When it comes to secondary structure of protein, you can write that this is the shape in which long polypeptide chains can exist. So here also, if you are writing for the secondary structure of protein, you can also give the answer as coiled form of long polypeptide chains in alpha helix or beta pleated structure, because that is also a definition or a structure, beta plated sheets. So this is also an example for a secondary structure. For primary structure, only one particular point is there, that it is in simply amino acids which are linked in a particular specific sequence. Okay, secondary structure is a shape in which long polypeptide chains can exist. That is simply the answer. But if for that time, if you remember, like that secondary structure is basically in the form of alpha helix or beta pleated structure, if you are writing, still also you will get the marks, because that also is a definition for the secondary structure. But please make sure that don't write too much things, because they have only, question is only for 1 mark. So 1 marks means you should only mention one difference between both of them. So either keep with only thing, that means it is linked with only amino acids in a single chain, but the other thing, chains which are coiled into different shapes, that is secondary structure. So for 2 marks, be precise and concise in whatever you are writing. So this is a case study question. So one thing about case study question is most of the time you don't need to read this particular case study, because rarely only there will be answer from the case study to the descriptive questions which are given there for the 4 marks. So this is a 4 marks question, means be also concise in whatever you are writing here. So write any two functions. As the name suggests, two function means you just need to write two exact functions for them. So there are many functions of the carbohydrates that is given in the textbook. You can also write, for example, carbohydrates is basically fuel for any of the organisms. So I can write, can act as a biofuel. Also, you can also write it provides energy. Basically, biofuel as well as providing energy, same sentence only. But this can also be treated as two different points. As per the official answer key, these two are considered as two different points. So if you write both of them, that is also fine. Or I can write an instant source of energy. Basically, we are talking about carbohydrate. As a food source, we are also consuming all sort of carbohydrates. So think about any of that source of energy, or we can say energy storage. Or if you are thinking about other carbohydrates like cellulose, you can say cell wall formation. Or if you want to remember about all the possible carbohydrates, which can be like glucose also, you can write fructose or starch or any of the carbohydrates you can think about and write the prospective functions. You will be getting the marks for this one very easily. So that is the answer to any of them. So second question is what are monosaccharides? So here also monosaccharide, they have asked what are monosaccharides. So some mistake which students generally make it, they will directly ask the name, give the names of monosaccharide like either glucose, fructose or galactose. That will not give you marks. You need to give the proper wordings what exactly is monosaccharides. So monosaccharide is basically a carbohydrate that cannot be hydrolyzed further into simpler unit of polyhydroxy aldehyde or ketone. So as the name suggests, any carbohydrates. For example, if you go to any disaccharides or polysaccharides, when they undergo hydrolysis, that means whenever during their reaction with water, they can break down into smaller carbohydrates. That is about the disaccharides and the saccharides which are having more than one unit of monosaccharides. But monosaccharides, you cannot break them down into smaller units. That is the smallest unit of carbohydrate available. So monosaccharide, you can write a carbohydrate that cannot be hydrolyzed further into simpler unit of polyhydroxyaldehyde or ketone. Or also you can also write that simplest unit of carbohydrate which cannot be into smaller units of polyhydroxyaldehyde or ketone. So simply those who are wondering what exactly is polyhydroxy aldehyde or polyhydroxy ketone means, that is another way to say that they are saccharides or carbohydrates. So carbohydrates are basically polyhydroxyaldehyde or ketone. So if they cannot break down a particular thing into smaller units of this polyhydroxyaldehyde or ketone, that means that is the smallest unit. So you can write any of this, you will get the marks. Now for the or question or the C question, C part of this question, write the name of deficiency diseases caused due to the lack of vitamin D. So you can write any of the disease. Basically, there are two particular disease. First one is rickets, and this is basically for children. So if you write rickets, you will get the marks. Or you can also write osteomalacia, which will be osteomalacia, which is basically for the adults. You can also write that also. You will be getting the marks for any of them. This is or any of them, because there are no other diseases deficiency which are caused by the deficiency of vitamin D in your textbook. So if you write either rickets or osteomalacia, you'll be getting 1 marks. So if you want to see the distribution of marks for all of this will be 2 marks, any two difference. So basically you'll be getting 2 difference for each of them. You'll be getting 1 plus 1 marks. Now for second time, you need to give the difference. I mean, what exactly is the hydrates? This will be 1 marks. And C part, whatever you are writing, you will be getting 1 mark. So simply you can get the 3 marks from this one. Now, if you look at the third one, that means C or question from here. C, first one, that is name the hormone which maintains the glucose level within the narrow limit in the blood. That is very easy. That is insulin. So that also gives you the answer. So that will also give you 1 mark. This is our question. So whichever you feel is correct, don't need to write both of them. You will not get correct marks. Here also, one thing you need to be careful. Or means when whoever checking the paper, they will only look at the first part of the answer which you have given. For example, if you have given C as Ricketts answer here, and if you again answer C as insulin, the first answer which you have given, only that will be taken into account. So be careful when you're attending questions like this. So I hope it is clear. Now this question, which of the following statement is not true about glucose? First of all, it is an aldohexose. That is correct only because 6 carbons are there. It also contains a carbonyl group. So we can say aldohexose. On heating with HA, it forms N-hexane. That is also correct only. When you are heating glucose, so when you are heating this particular glucose, what will happen is with HA, it will react to form a 6-membered chain of CH3, CH2, 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 CH3, which is N-hexane. So that is also correct only. It exists in furanose form. That is actually incorrect. Glucose actually exists in some form of pyranose because, see, this membered thing is called the furanose form. This is called furan. So furanose form is only for fructose. As the you can derive from the furanose has F, fructose also has a furanose, fructose. When you're pronouncing these two words, it will be clear that both of them have F in them. So you can say furanose is actually a form of fructose exists in the form of furanose. When it comes to glucose, it is pyranose. So how is that form? Is basically, so glucose exists in this type of molecules. For example, this one is, this is the this is basically the, this is called glucose pyranose form, also called as, this one is called alpha D glucose. So this is a form of glucose. So this means, see, throughout this chapter, one thing if you want to remember is two key things are there. Reactions of glucose. The question, we will see the question, reactions of glucose as well as the structure of glucose. So if you know both of them, it will be very easy to answer most of the questions. Okay, so structure of every glucose will be having 6 carbons, where one end is CHO, other end is CH2OH. And it will react with lot of compounds. HCN is there, NH4OH is there, bromine water is there, as well as HCN is there. So different cases, different products will be formed. So all of them are important. So in your textbook, these things are there. If you keep on this thing itself, you can sure short get marks from this one. So finally, it does not give shift test. That is also true only. Shift test is basically given by the aldehydes. But the problem is glucose does not always exist in this particular open chain format. It will be existing in a closed chain format as well. That is another existence. It's also shown. So that is called alpha D glucose as well as beta D glucose. So it will not give the shifts test always. So now for the next question, this is an assertion reason question. One thing most of the student makes in assertion reason question is directly jump into the answers. So before that, to solve any assertion reason question, first thing you need to do is read the assertion properly. All naturally occurring alpha amino acids except glycine are optically active. Now, before reason statement, you just add a statement because, just a clause, because. So will this term, I mean, because most naturally occurring amino acids have L-conflicts, will that satisfy whatever you are saying or not? That is the question. So here, all naturally occurring alpha amino acids except glycine are optically active. That is correct only. Optically active means we'll be having a carbon which will be surrounded by basically 4 different groups. So in case of every amino acid, on one side you have amino group, other side you have COH group, and one hydrogen. So glycine here one R group here. That is this is a common form of amino acids. Every amino acid has this common form. Now, when it comes to glycine, this R is equal to H for glycine. So that means glycine will never have the 4 different groups. It only have 3 different groups around this particular carbon. That means that is not a chiral carbon. Chiral carbon means which have 4 different groups in a attached to a single carbon. So glycine is not optically active, but when it comes to all the remaining amino acids, this R group can be different. If you look at the tryptophan or tyrosine or any other amino acid, if you take, this R group will be different. So that means except glycine, everything else is optically active. So this statement is correct only. Now the explanation, because the explanation which should come is because glycine has 3 similar groups, something like that they can write, or they have no substituent group in the amino acid. That are all correct statements, but they have given is because most naturally occurring amino acids have L configuration. That is incorrect. First of all, that is incorrect, and that is also not correct explanation for the assertion. And another thing why it is incorrect is because most naturally occurring amino acid have here L configuration, or we can say LAVO configuration. They are telling, but it's not LAVO configuration. It is dextro configuration. It will be having. Okay, or we will be saying D configuration will be the default configuration of all the amino acids. Okay, so the answer for this question is assertion is true, but reason is false. So in almost all the board question, first option A will be assertion and reason true, but reason and reason is correct explanation. B1 will be assertion and reason true, R is not the correct explanation. C will be assertion true, reason false. So this is the way. And the fourth option generally will be assertion false, reason is true. So you need to careful about what are the options which are given there, and don't jump into conclusion when it come to this type of question. Now for this question, write the reaction involved when D-glucose is treated with the following reagents. So this is similar to what I have already told you. The reactions of glucose with these compounds are important. Here also, if you just write that exact reaction which is given in the textbook, that is in this format, CHO, glucose is CHOH 4 times and ending with CH2OH. So when you are reacting with HCl, here mechanism and all you don't need to worry, but still I will show you. This carbon has C double bond O H. Okay, so remaining these groups are there. So this CN minus from the HCN, it will attack here, and this reaction will happen something like this. But mechanism and all you don't need to worry. So what will happen is CH will be there, one CN will be there, and OH. Here one electron I told you, oxygen is having one extra electron. So this will absorb some water or H plus from somewhere in the surrounding, and it will get CHCnOH. So we call this one as a cyanohydrin. Even if you don't write the name, this is not compulsory. If you are writing, that is fine. If you are not writing, that is also fine. If you write just this reaction, you will be getting 1 mark for sure. So this is for A part. Now B part also same thing, which we have seen in one of the previous questions. CHO, CHOH 4 times, CH2OH. So when you are reacting with bromine water, what will happen is CHO will change into COOH, CHOH 4 times CH2OH. So this is called gluconic acid. So even if you don't write this name, still fine. If you write this reaction, you will be getting sure shot 1 marks. So I'm again repeating the same thing. When you are writing the answer, whatever given in the NCERT, that is the key point. And important thing about this particular chapter is glucose. All the 5 to 6 reactions that is given in the textbook, that is completely important. They will ask directly from there. So try to mug up or by heart or write yourself, practice with that particular reaction as much time as you want, but make sure that you memorize all of that single reactions. So this is a 3 marks question. So here also 111 means you just need to write whatever that is asked. What is the difference between native protein and denatured protein? So here also just 1 point will be more than sufficient. So when you're writing also, there are many other points for the denatured protein as well as native protein. So when you're writing also, please make sure that you write exactly to the point. Okay. So what exactly is the native form? Okay. So native form of protein. So native form means proteins found in biological system with a unique three-dimensional structure and biological activity. Okay, so this is the native protein. So native protein is any protein's natural form. Simply speaking, it is a natural form of any protein, but you should not write that okay, this is the natural form. So this is first key point is it is found in every biological system. That is the first key point here. It is found in every biological system. Now, another important thing is since it is found in every biological system means it will have its own property. You think about it, any protein in our body, it will be having its own particular structure. No proteins need to be synthesized separately. So they have their own shape as well as structure. So that also you need to mention. It has a unique three-dimensional structure and it will be having its own biological activity because whatever the proteins which was there in your body, they have their own functions, right? So you can write their own functions instead of biological activity is the term that is not coming into your mind. You can write simply biological functions. So they have their own functions. They have a dimensional unique structure. They have a unique structure and undergo unique functions or they have their own biological functions. So anyway, you can write, but this key point should be there. What are the key points? I'll once again tell you, this is found in biological systems. First thing. Second thing, it has its own structure. You can write in three-dimensional structure, but please don't write two-dimensional structure. That is not possible. So it have its own three-dimensional structure. That means globular protein, fibrous protein. You have studied all the tertiary structure of the protein. So you can write that and they have their own biological functions or the biological activity. So these 3 points should be there, then that will be correct only. But next, you need to write what exactly is the denatured proteins. So denatured protein means exactly the same thing only. So when this native protein, so when native protein subject to, for example, it is any change we are doing into native form, we will get into denatured form. So what change, how we can make this change? So this change can be subject to physical change like temperature or chemical change like change in pH. So whenever native protein is subject to any of these changes, what will happen is the hydrogen bonds disturbed get disturbed. Basically, whatever the hydrogen bonds holding all the tertiary structures and all, they will actually disturb. So what will happen is, and protein uncoils and unfolds, resulting in loss of its biological activity. So this is the key point. So this is called the denatured protein or denaturation of protein. So that is what they have asked. Denatured protein means any native protein which are subjected to physical or chemical change. Simply, if you write that, you will get the marks. Now, if you are not writing that, if you are writing any other thing, or you can write primary, sorry, I can say secondary and tertiary structure of native proteins, or I can say when secondary and tertiary proteins structures of native proteins are destroyed by some physical or changes, then such proteins are called denatured proteins. So here you don't need to write every single one of these sentences. So what you need to write is key point only you need to remember. So here, subject to physical change like temperature or chemical change like change in pH. So instead of writing change in pH and change in temperature, you can also write when it is subjected to change in temperature or change in pH, what will happen is protein uncoils, or we can say helix get uncoiled. Here also I can write another point. Helix get uncoils, and as well as the globules, which are the tertiary structures, they will also get unfolds. So you can write that sentence in any way. Protein uncoils as well as globules unfolds. It will lose its biological activity. That are the 3 points are it. Denatured protein. So if a question comes, this is the key point. When native protein subjected to physical and chemical change, its biological activity losses, it will form denatured protein. That is one way to say it. Or another way to explain this one is when native protein subjected to temperature or change in pH, it will uncoil its chains and globules get unfolds and you will be losing its biological activity. You will get the denatured protein. Or you can write secondary or tertiary structure of native protein destroyed and it becomes simply a primary long peptide chains by some physical or chemical changes. It is called denatured protein. So simply you can write any of this thing, but key point should be there. Subjected to physical and chemical change, that one point should be there, absolutely. And second point is protein structure get destroyed. You can either write in words like it uncoils or unfolds. That is one way, or you can write secondary and tertiary structure get destroyed and only primary structure remains. Okay, so these are the 3 key points. If you write any of these 3 points, you will get the marks. Now the next part is which one of the following is disaccharides. So what are disaccharides? So here disaccharides will be having 2 monosaccharides. So let me again go back to the previous one. So previous one, what is the difference between they have? So instead of this, based on the answer, you only need to write, if you write just 2 points or 1 point, 3D structure intact and biologically active. Simple words. If you just want to write simple words, this is one. And this one denatured means 3D structure destroyed and biologically inactive. If you just write these 2 things, simply you will be getting the 1 marks. That's it. But other things, if you are writing subject to this change and all, still also you'll be getting marks. Okay, so any one difference only you need to write. For native proteins, only one thing is they have biological system, they have a unique structure, and they have biological activity. Or if you're writing they have unique structure and biological activity, you'll be getting the marks. Or you can do they are found in biological system and they have unique functions, still you will be getting the marks. Denatured means when subjected to changes, it will lose its 3-dimensional structure and biological activity. That is correct. Or if you are writing their 3D structure get destroyed and biological activity is also completely gone, that is also correct. So now the next one, which among the following is a disaccharide? Just 1 mark question means you just need to mark whatever the disaccharides. So here disaccharide is lactose. So if you look at the other one, glucose is a monosaccharides, amylose is a polysaccharides, Fructose is also a monosaccharide. So the answer is lactose. Which vitamin is responsible for the coagulation of many times repeated thing, coagulation of blood? Whenever you are hearing that, coagulation. When you are speaking, pronouncing coagulation, what will come? The first word which you are pronouncing, first syllable on that coagulation is Ka. So Ka means vitamin K. You can remember it as vitamin K. So the answer is vitamin K. Just one left, you'll be getting the marks. So that is the answer to 7th question. Now for this question, which among the following are examples of denaturation of protein? So this is also something that is directly given in the textbook only. Clotting of blood is not an example for the denaturation of protein. Coagulation of egg white, curdling of milk, both of them are coagulations example. So answer will be option D. So denaturation of protein in the textbook, whatever the example or as well as the explanation which I gave in the last particular question is important. Second thing is what are the examples. So for example, question can also come, how egg white coagulation is an example of denaturation. So if a question like that comes, you can say that, so coagulation egg white, when you are heating egg white is basically protein only. So when you are heating egg white, what will happen is temperature, physically you are changing the proteins, native protein. So that means it will destroy the 3D structure of the protein or the secondary and tertiary structure, it will get destroyed. Coils, helix get uncoiled and globules get unfolded, and you'll be getting with a single polypeptide chain. That means the protein's activity is completely destroyed. So that is also you can answer for a 2 marks question. Ninth question, nucleotides are joined together by. So here remaining options also we can check. Glycosidic linkage. This is between the carbohydrates or disaccharides or the polysaccharides. Peptide linkage, that is basically between the amino acids. Now hydrogen bonding. So hydrogen bonding is between the polypeptide peptides or amino acid change or proteins. The secondary structure as well as the tertiary structure will be having hydrogen bonding along with some other linkages are there, disulfide linkages, van der Waals forces, different things are there. So they are all present in the proteins. So nucleotides are basically joined by the phosphodiester linkage. That will be from the nucleo. Basically, every nucleotides are joined together by phosphodiester linkage, where the sugar part, the deoxyribonucleic acid, that is DNA, or ribonucleic acid, that is RNA. So they have a ribose sugar or deoxyribose sugar is there. They have a nucleic nitrogen base is there. So these nitrogen bases plus this sugar, they are connected with the phosphate part, and this phosphate is actually linking both different nitrogen base or nucleotides. So this phosphodiester linkage is there. You don't need to worry about the structure and all. Phosphodiester linkage, what name, that only is important thing. So that is that, and remaining things also keep in mind, right? Glycosidic carbohydrate, peptide means amino acids. 10th question, scurvy is caused due to the deficiency. So this is simply deficiency question. Scurvy is basically caused by vitamin C or ascorbic acid. So remaining things are also there. Vitamin, for example, what are the deficiencies of other things? That is also important questions I have asked before. So that is also important. What are the deficiencies? For example, vitamin B1. Vitamin B1's main deficiency is beriberi, which means simply we will lose our appetite as well as retarded growth. That is basically beriberi means. And vitamin B2 or riboflavin. So this will be having simply anything that is related to eating or basically eating or digestive systems problems. They are all actually due to this one. One example that is given in the case of chelosis or chelosis. And glutamic acid. What is glutamic acid? So glutamic acid is actually not a vitamin. This is basically a amino acid. So whose deficiency we don't need to worry. So this is don't need to worry. So remaining things just keep in mind that it can also ask for other questions. Now 11th question, vitamin D cannot be stored. This is also assertion reason. So assertion reason, same idea only we need to keep in mind. Will assertion, actually reason is the because statement. Is it correct or not? Is first thing is vitamin D cannot be stored in our body. First of all, assertion itself is false. That means you don't need to worry about the remaining options. Answer will be option D, where assertion is false. The reason is true. Vitamin D is fat soluble vitamin and is not excreted from the body in the urine. That is correct only. So if the question is vitamin D can be stored in our body because vitamin D is a fat soluble body vitamin and is not excreted from the body in the urine. If they have given can instead of cannot, then this answer for this question will be option A. But here, since assertion is false, our answer will be option D. Now for 12th question, next question, define the following terms. So defining means exact words only you need to write. Glycosidic linkage means what? It is the linkage between carbohydrates. You can write that as it is, you will be getting the marks. The next thing, linkage, or you can write oxide instead of linkage. You can also write oxide linkage. That is also correct. Now B part, invert sugar. So invert sugar, what exactly is invert sugar? Define, you need to define. If you are writing just like sucrose, you will not get any marks. So what you need to write is invert sugar. Why sugar is called invert sugar? So you can write completely, you need to write hydrolysis of, see this is defined. So you need to properly write what is happening. So sucrose brings about a change in optical rotation from dextro. That means initially it is having a dextro rotation to levo. Optical rotation from dextro to levo products. And this product is named as invert sugar. So we can say when what is called invert sugar, the answer will be sucrose. So why it is called invert sugar? Because its optical rotation changes from dextro to levo or plus to minus. You can write anything, but you will be getting the marks. Okay. So hydrolysis, that key terms here are hydrolysis of glucose, sucrose, hydrolysis of sucrose, and change in optical rotation dextro to levo. So these are the key points here. Then you will be getting the marks. And this product is named as sugar. So even if you, so this key point only you need to keep in mind. Other thing, if you are not writing also, you will get the marks. So next thing, C part, what are oligosaccharides? So oligosaccharides means you can write directly that these carbohydrates that yield 2 to 10 saccharides on hydrolysis. So this is the exact words which you need to write. Oligosaccharides, don't go and write about the disaccharides as well as polysaccharides or anything like that. Here, keep to the point. Define. Define means you just need to define what exactly that thing is. So here, key point about oligosaccharides that yield 2 to 10 monosaccharides on hydrolysis. Hydrolysis means simply breaking the carbohydrates, will be getting smaller carbohydrates. So answer is just that point only. Yield 2 to 10 monosaccharides. Now this question, in the Howard structure of the following carbohydrate, various carbon atoms have been numbered. Anomeric carbon. Anomeric carbon is always this carbon, which is directly bonded to this oxygen. This oxygen is basically part of that chain only. So this oxygen, which is directly bound to the first carbon. First carbon is, this is, if you remember the structure, CHO. Basically, okay. HOH, C, OHH, C, HOH, and CHOH, and finally CH2OH. So this is the open chain structure of the glucose. Now, if you look at the structure of glucose, what you can find is, except this third carbon, all the other carbon has structure like this. So this fifth carbon, when it is attacking here only, you will be getting this closed structure. So this is the first carbon. So depending on this first carbon's OH and H orientation, we will be getting alpha as well as beta. So this one is alpha D glucose is there. Similarly, there is something called beta D glucose. So when you're drawing structure like this, so when OH is on top, this one is called the beta D glucose. If OH is here on the bottom, we call it as alpha D glucose. So this carbon, this is all due to the changes in the first carbon, which is directly bound to the first carbon. That is called the anomeric carbon. So answer is option A. Now for the next question. 14th question, what is essential amino acids? Why are amino acids amphoteric in form? So 2 marks question. This is a 2 marks question. So let's answer one by one. So first question is essential amino acids. So essential amino acids. So essential amino acid means simply, this means amino acids which cannot be synthesized within the body and need to be supplied through diet. So that is a key point here. This is the essential means we cannot get them from our own body. We need to actually supply them through a diet. So that is first part is done. Now next part is why are they amphoteric? So why they are amphoteric? You can answer in 2 ways. First thing is due to presence of both carboxylic and amino groups. That is also a good way to answer amino groups. Or you can also write it as in sweeter ionic form, amino acid can react with both acids and bases. So this is also correct answer. You can either say that it has both carboxylic acid and amino group, or you can also say that, or you can say it's sweeter ionic form. That means when you're actually adding this particular thing into water, it will actually form an ionic form where you'll be getting ammonium ion as well as carboxylate ion. NH2 become NH3 plus and COOH will become COO minus. So in that case, this NH3 plus can give the hydrogen and COO minus can take the hydrogen. So that means they can act as acid as well as the base. Okay, so that also you can write any one of them. So you'll be getting 1 mark if you write correctly any of them. This will be for 1 mark. And if you write the essential amino acid, you'll be getting 1 mark here. So that is about the 14th question. Now again, one more long. This question is there. Case study question. So case study question also, here also. Cases mostly you don't need to read the question completely, or the cases you don't need to read completely most of the time. Because so far in CBSE, almost all the cases they have given some paragraphs they will take from somewhere, but mostly that paragraphs might or might not contain the answers. Okay, so here what products will be formed when DNA is hydrolyzed? So first thing, DNA composed of what thing? A sugar is there, and a nucleic nitrogen base will be there something, and a phosphoric acid or phosphate group is there. When phosphate group is actually reacting with hydrogen, every phosphate will form phosphoric acid. every sugar, whatever the ribose or whatever it is there, they will form that particular sugar. Nitrogen base will be forming as base only. So here in the paragraph, some of things are there, phosphate groups are there, and all these things are there. So we will directly write like phosphate. That is completely incorrect. So basically, every DNA, when it is hydrolysis, it will be forming 2-deoxy ribose. Okay, only difference is in case of RNA, you will be getting ribose sugar, and here you will be getting 2-deoxy ribose. So that is already given in the paragraph, but mostly it won't be there, but here it is in the paragraph if you are confused about that. So this is a sugar. Second thing is phosphate from every DNA. That means phosphoric acid. Every we call it as, when we can say, nucleotides. When you break, nucleotides are connected through phosphodiester linkage. So when you break this nucleotide singly, you will be getting phosphate, sugar, nitrogen base. So when you break each of them separately, you will be getting nitrogen base, sugar, as well as phosphate. See, phosphate react with water to form phosphoric acid and any nitrogen base. Here you don't need to specify nitrogen, nitrogen or nitrogenous, basically nitrogenous base. That is also correct. You don't need to specify what uracil or thymine or adenine, guanine. You don't need to cytosine. You don't need to specify that. So that is the first part. How is this? Is you will be getting 1 mark. Okay, 1 mark you already got here. How is DNA referred with reference to structure? So structure you can simply write. We can say DNA double-stranded and RNA is single-stranded. Simply you can write that, you will get the marks. And B part differentiates between nucleotide and nucleoside. Here this is given in the particular thing. I mean paragraph itself it is there. When these purine pyrimidine derivatives are coupled with ribose sugar, they are called nucleosides. So nucleoside means when you are having only, or I can write nucleoside. What do you mean by nucleoside is sugar. I mean it can be pentose sugar only. It can be ribose or deoxyribose. Pentose sugar plus nitrogenous base, you will be getting nucleoside. Now when it comes to nucleotide, basically you will be having same thing, pentose sugar plus nitrogen base plus phosphate, or I can say a nucleoside plus phosphate, you will be getting nucleotide. So if you just write that, don't write actually ditto there, please you need to write like pentose sugar plus nitrogenous base plus phosphate. So you'll be getting another 1 mark for this as well. Okay. So next one, C. First one, mention any two functions. Two functions means only two things only you need to write down. Mainly nucleic acid only have two functions. Basically two functions. First thing is preserve genetic information, or I can say transfer of genetic information, that is all. And RNA is actually used for the synthesis of protein. So these are the two functions of every nucleic acid. So you just need to write that two of them, you will be getting the marks. So C part, OR part, what is the second one? OR part, if you look at the second one, what is the linkage which joins two nucleotides? We have already discussed, which is phosphodiester linkage. Okay. Nucleotide in RNA and DNA, only difference is uracil, which is present in RNA. And in DNA, you don't have uracil, but you have thymine. Okay. Nucleotide of RNA, but not in DNA. That is the question. If the question was in DNA, not in RNA, then the answer will be thymine. So that is the answer there. Now, 16th question. Name the type of linkage responsible for the formation of proteins. Just 1 mark question only. So you just need to mention that the linkage is peptide linkage. So B part, write any two differences between the DNA and RNA. So two differences means just two points only. So for example, DNA, and if I write RNA, so for example, first one you can say double. DNA is double stranded, RNA is single stranded. And when it comes to other thing, we can say it has 2-deoxyribose sugar. Here I can say ribose sugar. And here I can say DNA has thymine base, and this one has uracil base. So again, also that it actually it replicates, does not replicate. You can say does not replicate. Or you can also write whatever the difference which you know. For example, RNA is actually used for protein synthesis, DNA is not used for that. DNA is mainly used for genetic transformation, RNA is not mainly used for that. So you can write any two differences. So you can say any two. You can write either these two, or you can write this one, this one, this one, this one, this one. Any two difference. Don't write all of them. You will not get extra marks. Any two suitable differences means any two suitable differences. You will be getting the 2 marks. So I hope it is clear. Whenever any question comes, any two differences, any one differences, or write the difference between, you only need to write the one difference. Write two differences means write two differences. One thing students actually mistake is they write lot of the things for simply because they know that. No need to do that. Your goal is to maximize the time you are having. You need to try attend all the questions, not like leaving one or two questions. So when question asked for writing the two differences, write the two differences. So I hope that is clear. 17th question, another long paragraph they have given. So rather than going through the paragraph and studying that, you can actually go to the next one. Question, give chemical reactions to show the presence of aldehyde group and straight chain in alkane. So basically there are two reactions I told you. In glucose, you need to remember the reactions, whatever it is given. So first reaction, aldehyde group, we have already seen the reactions. So CHO, CHOH, 4 and CH2OH. So here, when you're actually to check, verify, I mean, if you can react with what you can say HCl, or you can also react with hydrazine, or I mean NH4OH, to see that, okay, there is not hydrazine, ammonium hydroxide. So if you react with ammonium hydroxide or HCl, you can see that there is a carbonyl group, but that will not test that it is a aldehyde. So if you're writing any of that or plan to write any of that, please don't write that. That is just for proving that there is a carbonyl group, not for aldehyde. To prove that aldehyde group, only one thing is there, bromine, water. So this will change this into gluconic acid, which we have already seen, CHOH, 4 times CH2OH. And second thing, straight chain alkane also we have already seen the reaction, CHOH, CH, CHO, CHOH4, CH2OH, which will react with HI. While heating, you will be getting N-hexane. This is also an important reaction. This proves that all the carbons are actually bind in a single chain. So these two reactions, I told you, all 5 or 6 reactions that they remember all those. Very simple to remember only. Okay. So B part. So as you can see, this is a case study questions are basically 4 marks question. Okay. So if you write, this is most probably this will be for 2 marks. Give two equations are there. This is for 1 mark. This is for 1 mark. Now, next part is anomers. Define anomers. So what are anomers? Anomers also we have already seen. B actually has 2 OR parts. We only need to write any one of them. So, so let's look at that. So here, define anomers. So here, exactly what is asked, that only you need to write here. So when the question is about dividing anomer, just define what is anomer. So anomer means what? Anomer is basically in cyclic structure, or you can write in the pyranose form of glucose. In cyclic structure of glucose, differing in the configurations of C1 OH group, that are structures, cyclic structures of glucose differing in the configuration of C1 OH. That means here draw the structure of, you need to draw that, but that we will see for drawing. So here, if you see that, what exactly they are asking is, you can either write that this is simply anomer. Simply remember the definitions of anomer. Anomer are pyranose form of carbon, or if I draw it like this, for example, okay, so this is the pyranose structure, and here I can write HOH, HOH, OH, and again HOH, CH2OH, H. So what I'm talking about, this particular carbon, where actually this carbon which is directly bonded to here, this is C1. So this carbon, whatever the changes when OH is actually coming here and H is actually coming here, that means this position, if they change, we will get anomers. So this is the different cyclic structures of carbon where the configuration at C1 is changed. Okay. Or so you can also write or stereoisomers, or you can also write stereoisomers differing configuration of OH at C1, or you can also write C2, because this is also applicable for fructose as well. If you look at the structure of fructose, so fructose also will be fructose actually, instead of pyranose structure, you will be getting furanose structure. So there also you will be seeing something like this. Oxygen, I mean, structure will be something like this only. So here, instead of first carbon, this is the first one, this is the second one actually. That is why here only one difference is there. OH, H, OH, H, CH2OH. So in case of fructose also, this carbon C2, that is the anomeric carbon. So here anomers, you can write any one structure, I mean cyclic structures of glucose differing in the configuration at C1 OH group, or you can write cyclic structures of fructose differing in the configuration at the C2 CH2OH group as the stereoisomer differing in configuration. Both of them are C. Okay, basically simply remember that stereoisomers differing at the structure or the configuration at C1 or C2. Okay, so that is the answer here. Now for this OR part question, what they have asked is, draw the structure of beta D glucopyranose. So beta D glucopyranose means, I will just erase this thing here. Okay, this OH, when I'm writing this OH on top and H on bottom, this is called beta D glucopyranose. Simply, that is structure. So here, how do you remember this structure? That is the first question. So 1, 2, 3, 4, 5. See, except this third carbon, all in other cases, hydrogen is, or mostly in almost all other cases, hydrogen, except 3 and 5, all other cases, hydrogen is on the bottom. HOH, 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 CH2OH. That is the beta D, easy way to remember. And C1, if it is alpha D, first carbon will be having changes, but third carbon always OH will be on the top. And fifth carbon and all, you don't need to worry, their positions are fixed. Only third one, always it will be on, OH will be on the top. And first one, OH top means beta, OH below means that will be alpha. So next one is, sucrose is known as invert sugar. Explain. That is the question. So this product C part, we can write. So here, what you mean by invert sugar? That idea is, when you're actually hydrolyzing sucrose, it will actually changes the rotation, optical rotation. That means whatever, when you're actually passing the sample through a polarimeter, it will change the optical rotation from one particular direction to other direction. Initially it was positive, it will change it to negative. So this particular products are called invert sugar. So we can say hydrolysis of dextro sugar sucrose brings a change in optical rotation to levolotatory. So the product of hydrolysis is invert sugar. So you can write this in many different way. So just key point only you need to remember. Hydrolysis of sugar sucrose. So it will change its optical rotation from dextro to levorotatory. Or you can also write dextro means positive to negative. Or the optical change in optical rotation, or simply you write rotation, because in chemistry there is no difference in rotation means optical rotation only. So this product of hydrolysis invert sugar. So 3 points. Hydrolysis of sucrose, change in optical rotation from positive to negative or dextro to levo, product is called invert sugar. 3 points. Hydrolysis of sugar, optical rotation change, product is invert sugar. All right. So next question, which is pyranose ring of okay. So 18th question, pyranose ring of glucose is formed due to the reaction between C1 and C5. This I have already shown you that particular diagram. CHO, CHOH 4 times CH2OH. So this is the structure of glucose. This I can also draw it like this way. CHO, C, 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 2, 3, 4, 5, CH2OH. So here H, OH, OH. So this is 1, 2, 3, 4, 5, 6. We will always start numbering from the functional group. So this fifth carbon, when it is attacking here, that means here I will be having C double bond of H. So this attacking will result in the formation of pyranose ring, which will form something looking like this, which can form HOH. This is the first carbon, second carbon, third carbon, and the fourth carbon. This is the fifth carbon, and CH2OH, finally sixth carbon. So this is the pyranose form. So attack will be happening from C1 to C5. So that is the answer. Now 19th question, how do you explain the following? So this is also same question. So this is what I was telling. From hydrocarbon chapter, there is no new questions. Questions will only keep on repeating and repeating. So if you look at the previous one of the class videos, previous one of the question, you will see that to prove the presence of aldehyde group, we just need to react the same thing. CHO, CHOH 4 times, and CH2OH, which will react from bromine water. And what will form is a gluconic acid, which will be looking like this. CH2OH. So what you need to remember is this is basically glucose. So glucose, what is the reactant? That only you need to remember. What is the reagent? That only you need to remember. Bromine, water. Presence of 5 OH group. So this how will you prove? So this is a new thing, but here also reaction is very simple only. CHO, CHOH 4 times CH2OH. So what we will be reacting this one is acetic anhydride. So acetic anhydride, what it will do is it will react only with the alcohols. So what you will be getting? CHO, C double bond O, CH3 and CH2. This will be 4 times because 4 OH groups are there. This will also form C double bond of CH3. So in case those who don't know acetic acid, acetic anhydride, so this will be C double bond O, C double bond O, CH3. This is acetic anhydride. So when you're reacting, what will happen is all the OH group, this will actually attack here, and this reaction will happen like this, and this will go, and this particular bond will break. So you will be getting OCO, CH3 bond there. But just remember that these are the two marks question. This is 2 marks. So if you write to both of them, you will be getting the answer. So this is also another easy question. So question number 20. So this is also something we have already explained. See, define the following peptide linkage. How will the peptide linkage form? So peptide linkage, you can define directly. This is the linkage which joins amino acids through C double bond O NH bond. That's it. Now, second one, denatured protein, which we have already explained. Denatured protein means it is the native protein which is subjected to physical or chemical change. It will lose its 3D structure as well as biological activity, and that type of protein is called denatured protein. Simply, native protein without biological activity and without structure is called denatured protein. So that you can write there. Now, why do amino acids show amphoteric behavior? This also we have discussed. So amino acid amphoteric behavior is due to the structure. We can write that it has a carboxylic acid group as well as amino group. Or you can also write when it is Switter ion formation, when it is formed at Switter ionic formation. I mean, amino acid in its Switter ion form can actually work as and react with acids as well as bases. Or you can also write due to the presence of both carboxylic acid group as well as amino group. Now, C part, B part, you can write like this. C part, if you go to C part, first one, how can you differentiate between fibrous as well as globular? You can write any 2 points. In my opinion, go for the simplest one. Okay, simplest one is fibrous means basically insoluble, and globular basically soluble in water. Simplest point only, simplest difference only, insoluble and soluble in water. And if you want to explain further, you can say parallel polypeptide chains. Here you can say spherical polypeptide chains. Any one difference only you need to write, you will be getting the 1 mark. Now for the C or part, write the names of 2 different secondary structure. Basically, you know that secondary structures are alpha helix and beta pleated sheet. Simply, you can get the half. You will, if you see, let's say in this exam, let's say you only write this one, still you will get half marks. If you only write this one, you will be getting half marks. So that is a good thing about exams. You will never lose marks. Whatever you know, you should write that. Don't overwrite. That is the thing you need to remember. Don't overwrite. You can write whatever you know, you will get the marks. So answer will be alpha helix and beta pleated sheet. Now 21st question, again an MCQ. Alpha helix, that is just secondary structure we have already seen. Assertion reason, vitamin K can be stored in our body. So that is true or DEK. So basically DEK, right? K and A. You can write any way you can remember. I just remember it as DEK. Okay. Oh, so that means DEK. I mean DAKE. These are actually fat soluble which are stored in the adipose fat tissues. So that means vitamin K can be stored in our body because it is a not water soluble, it is a fat soluble. That means reason is false. So that means our answer will be option C. Option C says that assertion is true, reason is false. So answer will be option C. Now 23rd one, 2 strands in DNA are not identical but complementary. Explain why it is complementary or why it is Complementary means you can simply write that. Okay. Complementary means because of hydrogen bonding, because hydrogen bonds are formed between specific pairs of bases. Or if it is too much for you, can also write that it is complementary because adenine can only form bonds with guanine, and thymine can only form with cytosine with two double bonds, two hydrogen bonds, but other with three hydrogen bonds. You can also write like that. Is a long explanation, but simple explanation. Always go for the simplest answer. Simplest answer is because hydrogen bonds are formed by the specific nitrogen bases in both of them. So that is why it is always complementary. Okay. For example, adenine, ATGC. That is the combination. Adenine can only bond with thymine. Guanine can only bond with cytosine. So that complementary only happens because of the specific formation of the hydrogen bond. So next thing, when it hydrolyzes, what are the products? Products we already have seen when DNA hydrolyzes deoxyribose and a nitrogenous base and phosphoric acid. We'll be getting phosphate, but phosphate when you are hydrolyzing or phosphate when you are reacting, phosphate will change into phosphoric acid. So that is answer. 24th one. So here, what write the reaction of following. HCN we have already seen it will form COH, CN. Remaining will be same only CHOH 4 times CH2OH. So that this is called cyanohydrin. This also we have seen. This reaction will happen. Bromine water also we have seen. It will turn into gluconic acid, CHOH 4 times CH2OH. So this one, this thing, in case those who don't know, this is called acetic anhydride. So this one is acetic anhydride. So this means this will only give only one product, that is CHO, CHO, C double bond O CH3. This will become 4 times and CH2 O C double bond O CH3. So these three products, I mean, starting with the acetic acid, you'll be getting 3 marks. 3 marks question for 3 of them. Very simple only. So that is the end. Okay.